Now we are here, 0. It is now. And going back to the past, we could clarify the environmental change through these 800,000 years. So temperature, this line, and the CO2 concentration, these lines, has been changed periodically. So the 10,000 years warm interglacial period and 90,000 years cold glacial period are repeated periodically during these 800,000 years. Then, what happened on the terrestrial environment? During the glacial period, almost all the bare ground in Antarctica were covered by thick ice sheet, except the top of some mountains with higher elevations. In interglacial period, with rapid warming, ice sheet started to melt and bare ground appeared along the coast area. So it is presumed that almost all the living materials and minerals were washed away during these 90,000 years glacial period. The thick glacial flow over the ground shaped the mother rocks, and you can find some striations. It is a scratch mark on the rocks by the glacial flow. Such shaping effect by ice sheet flow must completely destroy the past terrestrial ecosystem. It's a periodic resetting. So the lifespan of Antarctic terrestrial ecosystem is very short, just 10,000 years in maximum after the last glacial maximum time. So during these 10,000 years, they have to start from the environment without life, absolutely clear virgin field. Even now, it's after 10,000 years has passed from last glacial maximum, Antarctic terrestrial ecosystem is very poor. In shore station area, biodiversity is quite low. For the land plants, you can find only 7 species of mosses and about 60 species of lichens. That's all. No vascular plants here. And for the animals, the largest one is mite. It's only 3 species. So for example, do you know the species number of mosses in Peninsular Malaysia? Mohammed and Tan, 1988, published the checklist of mosses, where about 500 taxa were reported in Peninsular Malaysia. But in Antarctic, around the shore station area, it's just 7 species. Very poor environment. So this is the reason why many biologists were working in marine ecosystems. And terrestrial members are just a small number. I think the low biodiversity on the terrestrial environment makes the low biomass of biologists working on the Antarctic terrestrial ecosystem. But now, I want to show you our trial to draw a big picture on the history, transition of Antarctic terrestrial ecosystem after the last glacial maximum. So Antarctic terrestrial ecosystem is a quite unique environment to study the primary succession, because we know the starting point. It is 10,000 years ago. And gradient of exposure the gradient of exposure history must be recorded along the retreatment line of glacier on the ground. Then, if we can add the record of the successional change of ecosystem structure along the gradient, this gradient, the transition history should be totally reconstructed. So it is a very interesting challenge. So at first, I want to show you our trial to detect the time ruler on the bare ground. The glacier became thinner and thinner, altitudinally, and retreated horizontally. So when the glacier is retreating, two axes is present. Thinner presents the vertical altitudinal axis and the horizontal retreating axis. So the surface exposure age is not a simple and horizontal line. This vertical thinner procedure is very effective for making a timeline on this slope. So we try to clarify the surface exposure dating by using cosmogenetic nuclides, beryllium-10 and aluminum-26. We set up the target area in Scarborough, the largest ice-free area around in the vicinity of Shore Station. So on this ice-free area, we can set a 10-kilometer long range of retreat of ice sheet from west to east is expected. So in this figure, this is an ocean, and this is a bare ground scarboroughness, and here it's the ice sheet. So now the end line of the ice sheet is present here, but 10,000 years ago, this line should be over there. Then after these 10,000 years, this line is retreating from this direction, from west to east. So then Dr. Suganuma, our colleagues, and his friends tried the analysis. So this is a figure from the Kawamata et al. 2020 Quarterly Science Review. So this is the first result. The orange arrows, these orange arrows show the orientation of striations, scratch mark on the rock made by glacial flow. So it means the glacier was flowing on this bare ground from this direction, around there, east to west. So ice sheet was flowing from almost east to west over the ground, and sampling sites, surface exposure dating sampling sites are shown in this circle symbol on the ground. And the age of the surface exposure dating are shown with orange and blue values in these pictures, but it's very small, so I will show the next table. Okay, this is the surface exposure age plotted against altitude. The y-axis is the altitude, and separated into the northern side of scarboroughness and the southern side of the scarboroughness. So gray shade shows the timing of the ice sheet retreat in each area. So this figure shows the high elevation site, the exposure time is older than lower place. Okay, this line, the x-axis is 0 is now to the 5,000 years ago and 10,000 years ago. So it means high elevation place is exposed in the older times than low altitude region. So this very clear line is shown in here. It's not clear, but almost such a line should be drawn. And some data were very, very old. It means it is the top of the mountain, about 400 meters. It shows this top of the mountain summit was not covered by ice sheet during the last glacial period. So now we can understand the surface exposure period in this area, and their time scale is 6,000 to 10,000 years. The next step is to analyze the time record of successional change of ecosystem structure in each location with timestamps. But the terrestrial ecosystem is very poor, simple, so the ground sediments are very thin and no information in them. So we selected the target successional record, not at the ground surface, but in the lakes. Why we choose the lakes? The maximum surface ice thickness is under 2 meters in winter, even in winter. It means when the lakes are deeper than 2 meters, liquid water is present throughout the year at the bottom of the lakes. And in this liquid water layer at the bottom, biodiversity is quite higher than the surrounding terrestrial ground. So, as you see on this picture, the surrounding ground is very dry and like a rock desert, but at the bottom of these lakes, we can find prominent vegetation structure at the bottom, like this. Should be, I wish we can show the movies. So at the lake bottom, we can see some green vegetations. And you can see the structure. So these structures are composed with aquatic mosses, algae, fungi, cyanobacteria, and bacteria. The main component is cyanobacteria. And this cyanobacteria-based sediment is very thick, sometimes 2 to 4 meter depth. The sediment should show the history of the ecosystem of the lakes from the time when this area was exposed. Perhaps you can understand the richness of this biodiversity at the bottom of the lakes. It's very soft structure, and the main component is cyanobacteria. So we tried to catch up the sediment and analyze these cores. So this is a cross-section of sediment core from the lake bottom. You can see many layers with various colors. Most of these dark green and the orange color layer is composed of cyanobacteria. And this gray part is silt from glacier. And in some lakes, aquatic mosses are found in them. So by analyzing the structure, composition of plants, silt and carbon isotoping data, we could read the history of these lakes. So this is one of the examples of core analysis done by Mr. Koyama, our graduate student. We can read near to 8,000 years historical record from this sediment. Mr. Koyama analyzes 16S ribosomal RNA and clarify the bacterial diversity in these 3 lakes along the depth profile. So, for example, these lines are Lake Hotokeike Lake and left side is a deeper part, about 3-meter long sediment core, and the right side is the surface of the sediment core. It is almost the same in the other 3 lakes. So this is old lakes memory, and this right side is a now growing vegetation at the bottom of the surface of the lake sediment. And Koyama reconstructs the successional change of the ecosystem structure in these lakes. So depth profile means time series from some 1,000 years ago to surface vegetation, like this arrow. You can recognize some successional change during through 1,000 years. It's very preliminary data from small number of lakes, but it indicates clear possibility to clarify the successional change in the lakes. So now, we have only 3 plots of lake data in this area. These 3 green dots show the 3 lakes, as I shown before. And these 3 lakes are situated in the southern part of Scarborough, and on this right side figure, this plot was around here. The elevation is almost the same. So these 3 data is not so clearly representing this history of this Scarborough region. We have to choose many lakes along the altitudinal gradient and the horizontal gradient on this ice-free area. We have to work hard to check the lakes and collecting samples and analyzing sediment cores. So by selecting such a suitable lakes along the time rule, the altitude and horizontal axis, then we can reconstruct the successions of terrestrial environments after the last glacial maximum completely. It's our goal. So the Antarctic Terrestrial ecosystem is very poor, looks like a rock desert. But now, your image must be changed drastically, I hope. So this system is telling us the unique dynamic history of succession and colonization of our Antarctic ecosystem. We can find this history from the bottom of the lakes. And now I have more time, so I want to introduce one more topic concerning the lake vegetations. As shown in the previous figure, around here, the lake bottom bacterial vegetation has changed drastically. In this lake, this point, and in this lake, also this point. These 3 points of the changing period are almost the same age. It's about 2,000 years ago. This timing is very interesting for me because in some analysis of the sediment core before, Seto et al. 2002 reported the transition of one lake from the marine inlet to the ground saline lake to the freshwater lake. This transition spent 6,000 years old to now. So the environment of this lake changes drastically from marine to freshwater lake. From the sediment, we can detect the mosses, aquatic mosses, from this timing. From this timing, aquatic mosses have appeared, and now we can find many rich vegetation at the bottom of this lake, these aquatic mosses. So this timing is also 2,000 years ago. So this 2,000 years ago is a very important timing for the Antarctic lakes. The bacterial diversity is changed drastically, and the mosses appears at this timing. So I think this is the timing of this moss pillar making mosses are invaded in the Antarctic lakes. So this figure shows the moss pillar. It is a structure of mosses growing at the bottom of lakes, about 80 centimeters high, and standing at the bottom, all over the bottom of the lakes. And core analysis tell us that such rich ecosystem started just 2,000 years ago. 2,000 years ago, it's a time when Jesus Christ was born in Bethlehem. So I'm wondering, what happened during such short time, just 2,000 years? And this time scale also contains another drastic change on this planet. Industrial revolution. We are trying to detect the stigma of Anthropocene in Antarctic lakes. So I'm very happy if some young researchers or students watching this session will have an interest in the Antarctic terrestrial ecosystems. How do you think? It's very interesting, you know? So thank you very much.
Thank you very much, Professor Satoshi Imura, for a very interesting talk and about the transition of Antarctic terrestrial ecosystem. It's indeed interesting to know how the changes are occurring over the years. And I wonder how this would be coming in coming years, and how now you mentioned the industrialization and all the changes that happen, and the changes have been slow. Maybe as we're going on, the changes might be much more in coming time, and how that would be affecting globally as the changes are occurring at the core of these lakes. Okay. Okay, I think do we have maybe a few minutes for any burning questions anybody would like to ask? A very burning questions at the moment before we. Is a question in the chat, Professor, from Dr. Hio Chong Chin asking, I wonder what drives the ecological succession.
I think it is a input from other continent by airflow. Many diasporas are flying over the Antarctic Ocean from the other continent, drop down to the surface of the Antarctic bare ground. But the bare ground is very dry, so they cannot survive. But in the lakes, the very suitable condition is present at the bottom. So many of these diasporas are having habitation in the Antarctic lakes. Then the lakes become very rich and changing through the time, I think. So.
And there is another, is following up his question, is the succession sequence the same in all the lakes you examined?
No, only the small number of lakes we examine. So we have to make more samples and more analysis covering the wide area along the time scale gradient. Yeah.
Indeed. Thank you so much, Professor Satoshi Imura for such an interesting talk and insightful talk on this area. And I think Professor has opened a platform for the younger researchers to look into something different. Yeah, for those of you who are listening to this, maybe you can contact Professor to collaborate on and embark on this journey. I thank you so much, Prof. And I would like to invite everybody to remind for the next session. Yeah, our next OLAR session will start soon at 2:45. Yeah.
Okay. Thank you, everyone. Thank you, Professor Imura.
See you at the next session. Thank you, everyone. Have a good day.
Thank you.